Hello, welcome everybody. On behalf of Biochemical Society and Portland Press, I am pleased to welcome you to this webinar. This webinar is part of the Biochemistry Focus webinar series. And topics of the series include different research areas in the molecular biosciences, as well as practical sessions to support career development. Now, each webinar will give you the opportunity to ask questions via text, and we welcome suggestions for future topics and speakers for the future webinar series. And please look at the Biochemical Society website for more details. Now, I am Aga Gambus, and I'm a researcher for University of Birmingham. My lab studies regulation of DNA replication using through the signaling by ubiquitin and sumo modifications, which is very appropriate to the talks that we are going to hear today. This particular webinar today is part of our dedicated early career research program of webinars. And we are really delighted to provide this opportunity today for Ivan and Ben and Eszter to share their work with the molecular biosciences community. Now, today's webinar is entitled New Advances in DNA Structure Repair and Protein Quantification, and it reflects the subjects that the three short webinars are going to be presenting. Now, before I hand over to our first speaker, who's going to be Ivan, I would like to remind you that you can ask questions throughout the whole presentations by writing the questions down in the question box, which you can see on the side. And the questions will be asked at the end of all of the three talks, so we're not going to pause between the talks for the questions, but they will all be asked at the end, and we will do our best to ask as many questions that you submit in the question box as we can. And please, when you write the question, state at the beginning who the question is for, so we can equally divide questions between different speakers. Okay, so that's the organizational stuff done. So let's start with our first speaker. So our first invited speaker today is Ivan Corbeski from University of Zurich. Ivan is a postdoc in a Cavendish group at the University of Zurich and is working on the mechanism of proteins involved in epitranscriptomics. I love the name epitranscriptomics, and which can potentially act as targets for anti-cancer therapies. Now, Ivan gained his PhD at Utrecht University in 2019, and now his postdoctoral research is focused on structural and functional characterization of histone chaperones, and he determined the first structure of a histone chaperone bound histone octamer. And today, Ivan is going to present this work to us. And as you can see, the title of his presentation is Chaperoning of the histone octamer by the acidic domain of the DNA repair factor APL4. Ivan, it's all yours.
Great. Thank you very much, Aga, for the kind introduction, and thanks to the Biochemical Society for this great opportunity. Welcome to all the biochemists watching from around the world, and I want to start by saying chaperone, not just because it's my favorite word in science, but I think it also has a very interesting origin from social life, which I want to bring closer to you. The term chaperone actually comes from the Latin capa, later French chaperon, which means simply a hat that was worn generally by people who looked after good behavior in public. And later the person itself who was wearing that hat was called a chaperone. So the original definition of a chaperone is a person that accompanies and looks after another person or a group of people. And to illustrate this a bit from an example on our current social life, chaperones are also simply parents who look after their children, like for example in the amusement park when they want to ride a roller coaster, because in the absence of a chaperone, kids like to do crazy kinds of things like riding the roller coaster upside down, which is dangerous. So Chaperone makes sure that they get properly and safely onto the roller coaster of their lives. And with this little excursion into the meaning of chaperone, we can now go together to the, of course, molecular chaperones that I want to talk about today. And all we have to do is to take this roller coaster and shrink it. Make it real small until it's as small as a cell nucleus, and then place it inside that nucleus, and we arrive at our genome in the form of chromatin, the roller coaster of life. And at the center of it lies the nucleosome, which is the fundamental repeating unit of chromatin. And what I would like to call the children of chromatin are the histone proteins, which form dimers and tetramers through a so-called handshake motif. And the general model says that one H3H4 histone tetramer can bind to DNA to form a tetrazome, to which two H2H2B histone dimers can bind one after the other to form first a hexasome, and ultimately the nucleosome is formed with the DNA wrapped around the histone octamer. And the chaperones in chromatin are the histone chaperones, and there's a whole army of them. And what they do is they chaperone the histones in the process of nucleosome assembly and disassembly, which will be the focus of my talk today. The question is, how do they do it? What are their interactions with the histones, reaction steps during the process, and what are intermediates involved? And there are different models of nucleosome assembly, and a well understood one is from in vitro dialysis, which is started at a high salt concentration at 4 degrees, because the histone octamer is stable in these conditions and it dissociates under physiological salt and temperature. The ions help to prevent unspecific interactions with the DNA, which can then bind with the highest affinity to H3H4 first, as the salt is dialyzed out, to then wrap slowly around the histone octamer as the salt is reduced to physiological levels, and to form the nucleosome, which is then also stable at room temperature. So for nucleosome assembly by histone chaperones, there are also models that look at the energetics of the reaction. And the first is a thermodynamic model. So without a chaperone, which is here to the left, the histones interact non-specifically with DNA, which leads to irreversible aggregation, making nucleosome assembly to the right basically impossible. And the archetypical histone chaperone NEP1, which this model is actually based on, binds the histones with very high affinity to bring their free energy closer to the nucleosome, and that makes the assembly the energetically favorable reaction. And there is an alternative so-called kinetic model, and here it is proposed that histone chaperone binding leads to a stabilized transition state that reduces the activation energy for nucleosome assembly, that then occurs via direct deposition of the histones onto the DNA in a more stepwise manner. But the question here is, how might such an activated chaperone histone complex look like, and what could be the steps towards nucleosome assembly? And the chromatin process that we wanted to elucidate this on is DNA repair, which maintains genome integrity. And we focused on non-homologous end joining, which is the emergency pathway to repair DNA double-strand breaks. And during this process, chromatin is decompacted when there is a DNA double-strand break. The repair machinery recognizes the break, repairs it, and chromatin and the nucleosomes are compacted again. The question is, what happens with the histones during this process? How are they chaperoned, and how are the nucleosomes reassembled? And as it turns out, a central factor in the repair machinery is the protein APLF, which holds the machinery together as a scaffold, and it has histone chaperone function in its C-terminal acidic domain. So we set out to study the APLF acidic domain at molecular detail. And here is the APLF domain architecture. So it has at its N-terminus domains and motifs for binding DNA repair factors. And at its C-terminus, it has 2 zinc fingers for binding poly-ADP ribose, which is a signal for DNA damage. And at the very C-terminus, it has the acidic domain that binds histones. And this domain is made up mainly of negatively charged residues, but also importantly, it has several aromatic residues, and its very C-terminus, it has a Kr motif that contains a few lysines and arginines. And we measured of this domain NMR spectra free, and when it's bound to the different types of histone complexes. And what these experiments showed to us is that APLF is intrinsically disordered, and it has two different binding sites for the two different types of histone complexes, and each binding site has a double aromatic anchor binding motif to stabilize the interaction. Double anchor 1 that is specific for H3H4 binding, and double anchor 2 that is specific for H2H2B binding. And mutating these aromatic anchors to alanines either strongly weakens or completely abolishes histone binding, as we show here by ITC. So two different binding sites for the two different types of histone complexes suggest that APLF could be able to bind both types of histone complexes simultaneously, which we then also tested by size exclusion chromatography under conditions under which the histone octamer by itself would not be stable, and that's why it dissociates into the two types of histone complexes. But when APLF is added to it, then one big and stable complex is formed. And by native mass spectrometry, we could show that the histones are present in this complex in the same stoichiometry as in the histone octamer, and there are two copies of APLF-AD bound to this complex. And in ITC experiments, we confirmed that APLF-AD can bind the histone octamer with very high affinity and, crucially, with a higher enthalpy and entropy of binding than the individual histone complexes, which indicates a synergistic histone binding mechanism by APLF-AD. And after these results, we were convinced that we're dealing with a stable complex, and so we set out to try and determine the structure of this, and we managed to get a crystal structure at 2.35 angstrom resolution of this complex, in which part of the DNA of the nucleosome is effectively replaced by two copies of APLF to form what we call the chaperonosome, in which APLF is fully disordered, and it traces the positively charged surface of the histone octamer with its aromatic anchors buried in hydrophobic pockets, mainly in the alpha-1, alpha-2 patches of H3 and H2B, and APLF binds the histones to interfere with DNA binding over both DNA gyres through both steric and electrostatic hindrance. Now, we determined the structure only of the histone binding domain of APLF, but we could also model in the Kr motif with the help of cross-linking mass spec data in two different conformations. But in any case, the two copies of APLF form a cradle in which the histone octamer is embedded, with most of the DNA binding surface of the histones covered by APLF, except for the region around the nucleosomal diet. And to show that this structure is a meaningful structure, that the interactions that we see are also happening in cells, we did some cellular studies, and in DNA damage tracks, we could see reduced recruitment of APLF when the aromatic anchors were mutated, actually a similar reduction as if the entire acidic domain was deleted. And in pull-down assays, mutations in either the H3H4 or the H2H2B binding sites led to reduced binding of both histone complexes. And this indicates that in the cells, the substrate for APLF might indeed be the whole histone octamer. We then tested in vitro what APLF could actually do with the histone octamer. And by using a short DNA fragment, we could detect a ternary complex between APLF, all the histones, and that DNA fragment, while with a longer fragment, we only detected nucleosomes. And that APLF can promote nucleosome formation. We also tested with an with an MNase assay, microcoagulable nuclease assay, and saw that with increasing concentrations of APLF, also the amount of nucleosomes that are formed also increases. And to validate this fact that APLF can form the nucleosome and the structure that we determined, we wanted to know more about the mechanism. So we performed an in vitro chaperone-based assay that is based on native page and detecting the DNA. So what you see here is when you take DNA and add an excess of histones to the DNA without a chaperone, this leads to aggregates which are removed from the assay, and hence there is no signal. But when you add to the chaperone APLF, aggregation is prevented and the nucleosome is formed. But when you mutate the aromatic anchors, there are less contacts between the chaperone and the histones, leading to more unspecific interactions between the histones and DNA, lower nucleosome assembly activity, and more aggregation. And when all 4 anchors are gone, the chaperone cannot control the histones at all anymore, which can then fully aggregate with DNA. And we complemented this assay results with ITC, and crucially, as determined from ITC, these mutants actually are still capable of binding the histone octamer, still with quite high affinity, like wild type. And this is due to enthalpy-entropy compensation, which is a strongly pronounced phenomenon for interactions with intrinsically disordered proteins like APLF. And this leads to only little changes in binding free energy when you introduce mutations. And together, these results mean that the conformations of the mutants that allow for unspecific histone interactions with DNA must also be possible in the wild-type protein, only then to allow for controlled interactions with DNA through competition of the aromatic anchors for histone binding. And this translates into a chaperone-mediated nucleosome assembly mechanism in which, under physiological conditions, APLF can bind the histone octamer to form the chaperonosome and prevent unspecific histone interactions with DNA, which can then interact with the only free binding site on H4 to then wrap around the histones as it displaces APLF, which can remain bound until the nucleosome is fully formed. And looking at the energetics, while, as I told you, chaperones like NEB1 can explain the thermodynamic model of nucleosome assembly, with APLF, we can now explain the steps of a possible kinetic model in which the chaperonosome represents the activated form of the histone octamer, which is ready to be deposited in a stepwise manner onto the DNA to form the nucleosome. And how can you remember this mechanism? You just have to think about the roller coaster. The chaperone hugs the children and then releases them one after the other and puts them onto the roller coaster of their lives. And now that I'm at the end of my talk, I would like to leave you with the final picture that I have from finding the chaperonosome, because I believe that we can learn a lot from it. So if you remember, I told you that the histones form dimers and tetramers through the so-called handshake motif. And whereas they're divided alone, I showed you the chaperone brings them together. And following this example, I think we should put all our hands together and bring the story together. And that was made possible by these people here. So I want to thank Hugo, who was my supervisor during my PhD, together with Rolf and Hans. We determined the crystal structure together with Tisha and Xiaohu. We did the nucleosome formation assays with Francesca and Bruna. The CELDA assays were done with Heiko, Case, and Wouter. The mass spec we did with Albert and Domenico. we also did some additional studies involving small angle X-ray scattering at the EMBL with Dimitri and Melissa. I did the ITC in the lab of Marcellus and Annaluz. And I want to also thank my current supervisor, who still lets me present my previous work, Amedeo. And the funding came from the Dutch Science Foundation, Marie Curie Actions, iNext, and Instruct Ultra. And with this, I want to thank everyone who was listening. And I'm looking forward to questions later.
Brilliant. Thank you so very much, Ivan, for incredibly interesting talk. That was very nice. And now I will act as a chaperone of this webinar and dive straight, move us into the next talk, which is going to be from next invited speaker, Ben Foster from University of Oxford. Now, Ben is a postdoctoral research associate at the group of Ian Gibbs Seymour at the Department of Biochemistry at Oxford. And he did his PhD training with Till Bartke, investigating the binding and enzymatic activity of chromatin reader proteins, with particular attention to the E3 ubiquitin-ligase, UHRF1. Now, Ben's research focuses on using protein biochemistry to investigate complex signaling pathways, especially ones involving ubiquitin. And the talk that he's going to present today is all about activation of the ZUP1 deubiquitylase by the RPA complex. Ben, it's all yours.
Okay, thank you, Aga, for the introduction. Thank you to the Biochemical Society for giving me the opportunity to share some of my work here today. And I'm going to present to you how the role of one particular binding partner, the RPA complex, sort of regulates the biochemistry and enzymatic activity of a recently characterized deubiquitylase known as ZUP1. So post-translational modifications, such as phosphorylation, polyubiquitylation, ubiquitylation play vital roles in quite a numerous range of signaling pathways, such as in DNA repair. And post-translational modifications by their nature need to be reversible. So while you have the addition of ubiquitin in various forms onto DNA repair proteins, as sort of shown here, you also need to have a regulated way of removing these after the signaling takes place. And for polyubiquitin chains or ubiquitylation, you have proteins known as deubiquitalases or DUBs, which themselves need to be regulated in an appropriate manner so they remove the signal once the signaling pathway is over. So DUBs can be classified into 7 different classes. So you have metalloproteases on the left and thioproteases or cysteine proteases on the right. And ZUP1 was very recently in 2018 by several groups identified as the founding and currently only member of this DUB class in the human genome, whilst there are orthologs in other organisms. So in previous publications, the crystal structure of ZUP1 was identified with ubiquitin bound at the catalytic cysteine. And this helped identify the catalytic triad that was present in this protein, but also it identified additional ubiquitin bound domains with that alpha helical ZUP1 helical arm and a motif interacting with the ubiquitin, here shown in orange. In addition, there's an atypical ubiquitin binding zinc finger that wasn't present in the crystal structure, but you require this ubiquitin binding zinc finger for the sort of maximal DUB activity of this recently characterized deubiquitylase. And from biochemical assays and DUB assays, we can see that ZUP1 is an endo-DUB in that it cleaves within a chain rather than from one end, and it seems to be highly specific for lysine 63 or K63 linked polyubiquitin chains. These need to be a particular length, so ZUP1 is much more active against much longer K63 linked polyubiquitin chains. In addition, there were quite a few cell-based assays that sort of identified ZUP1 as being involved in genome stability. So if you deplete ZUP1 in cells, you have increased susceptibility to DNA damaging agents, and you seem to have increased levels of replication stress, indicating a role for ZUP1 in sort of DNA repair pathways. So the function of ZUP1 in these DNA repair pathways is not entirely known. One way of getting more information was by looking for interacting partners, and this was done by several different groups using immunoprecipitation and mass spec. And one of these complexes that came down was RPA, or the replication protein A complex. So RPA itself is it's involved in DNA replication and a multitude of DNA repair processes. It's a single-stranded DNA binding protein, but it's quite quite high affinity or very high affinity to protect single-stranded DNA from nucleolytic cleavage that might be possible during replication repair processes. And this single-stranded DNA binding capability is brought about by various OB folds within the RPA70, RPA32, and RPA14 members of this heterotrimeric complex. It also has additional protein-protein interaction domains at the end terminus of RPA70 and a winged helix at the C terminus of RPA32. And these are required to recruit proteins to sites of DNA damage and for DNA repair. In addition, the RPA complex is quite extensively post-translationally modified. So at the end terminus of RPA32, it's quite extensively phosphorylated in response to DNA damage, but it can also be ubiquitinated and sumoylated as well. So how ZUP1 might bind to RPA is not really known. So I sort of arrived in Ian's group as a postdoc in Oxford to verify some of these interactions, including to the RPA complex, and see whether it combines the RPA complex directly, whether this interaction impacts ZUP1 or RPA function at all, and what the mechanistic basis, whether we can get some structural or mechanistic information of how this interaction might occur. So previous studies have sort of identified RPA as a binding partner by IP and mass spec, but from my protein biochemistry background, I decided to take a sort of different approach to purify and make the recombinant proteins from E. coli, so the human proteins, and just by size exclusion chromatography, this in the absence of any sort of DNA, you see an approximately stoichiometric one-to-one complex between the RPA complex and ZUP1, and this is equivalent whether you add single-stranded DNA or not, which I'll come on to in a minute. You also see interaction just by pull downs using an N-terminally tagged version of ZUP1. You see that it can bind to RPA in these assays. In addition, Martin, so he was previously a postdoc in Ian's lab, could see that could recapitulate a lot of ZUP1 interactions from human cell lysates. So here is just showing binding to RPA in the absence or presence of benzonase, again showing that the interaction seems to be independent of the presence of having single-stranded DNA present. RFW3 here is just a sort of positive control as a known interactor of RPA. So we can see that ZUP1 binds directly to RPA. It seems to be occurring in the absence or presence of DNA. Additional assays I've carried out in sort of bulk biochemical mobility shift assays show that ZUP1 can bind to RPA when it's already bound to a single-stranded DNA substrate, and also that RPA-ZUP1 complex can bind to single-stranded DNA when added as a sort of one-to-one complex. So ZUP1 seems to bind independent of DNA. Additional assays carried out by myself and also Martin from cell lysates is that ZUP1 seems to bind to phosphorylated RPA as well, whether you treat the cells with DNA damage and see the interaction with hyperphosphorylated RPA32 by IP, or if you use a phosphoromimetic version of RPA using purified protein, the interaction is still maintained. An additional sort of bit of information is that it seems to be independent of ubiquitin and sumo signaling, so you can add inhibitors to the E1 proteins that activate this sort of signaling just before cell lysis and carrying out the IP, and still see this interaction between ZUP1 and RPA. So, and I tried to work out which rings within ZUP1 were required for this interaction. I sort of made a suite of different truncations of ZUP1, and carried out GSD type pull downs from this. And to cut a long story short, we essentially find that there's a helix-turn helix immediately N-terminal to the catalytic domain, that if you remove this either by truncation or just deletion, you lose the interaction to the RPA complex by pull downs. And a similar sort of story is shown when you carry out these assays from cell lysates using flag IPs. So you see this helix-turn helix is required, but in what regions in RPA complex are the ones that bind to ZUP1? And because RPA is quite big, I turned to sort of a method I've used in the past, and it's already been mentioned in the previous talk, I think, is to use cross-linking mass spec. So to use a chemical cross-linker, in the case that I'm going to show here is with a lysine reactive cross-linker, and this sort of cross-links closely interacting lysines close together in 3D space, and essentially by processing these for mass spec and then using data analysis software, we can get an idea of which parts of proteins, whether within a protein or between a protein complex, are close to one another in space. And what we see here is that ZUP1 forms various crosslinks to regions on the RPA complex. So the winged helix of RPA30 C terminus into various different regions within the RPA70 protein within the complex. And this seems to be similar whether you have a single-stranded DNA there or not. And this is over various multiple samples as well, and you can map some of these crosslinks onto the known crystal structures and the vast majority of those fall within the maximum distance restraints of what you'd expect. A few are somewhat different that we're sort of looking into whether they are relevant or not for where the complex is formed. The other additional point is that we have cross-links both to this lysine 262 and lysine 522525 within ZUP on either side of this alpha 2,3, and it sort of places the RPA complex in close proximity. And we are trying to get more structural information either by cryo-EM or other ways to sort of map some of these onto a more 3D model of the ZUP on RPA complex. As far as within RPA, so I tried to map some of the cross-linked lysines onto known structures of the RPA proteins. So here on the left are the DNA binding domains within RPA 70, DNA binding domain A and B, and the crossing seems to be both outside and within the DNA binding groove of these OB folds, whereas in the context of other OB folds in the RPA complex, so DBD-C and DBD-D and RPA 32, these seem to be sort of outside of the DNA binding groove. As far as the common protein-protein interaction domains within the RPA complex, we see some cross-links to these as well. In the winged helix domain of RPA 32, these seem to be on the opposite side of where sort of canonical RPA 32 winged helix proteins bind to, which tends to be on the opposite side of where we see cross-links too. So whether that's relevant or not, we'll see when we get some more structural information on that. As far as verifying some of this, so previous master's student in the lab, Ryan, expressed the individual RPA complex subunits in E. coli, incubated this with tagged ZUP1, and could see that RPA 70 and RPA 32 on their own could bind to the ZUP1, sort of showing that the major interaction seems to be with these two members of the complex, and it's much more complex than just individual domains. I also tried to sort of delete some of these from the RPA complex to look at the binding to ZUP1, and whilst you don't lose it completely, there is a slight reduction in binding when you remove the common interaction domains of the N terminus of RPA 70 and the C terminus of RPA 32. So some reduction, but the other interactions likely play a role in forming this complex. So while I've been trying to get some more structural information on this, I wanted to look at the impact of this interaction on either RPA complex or on how ZUP1 behaves. So as I mentioned, I've looked at mobility shift assays or bulk biochemical assays, and ZUP1 binding doesn't seem to alter RPA's ability to bind single-stranded DNA. Something I've not looked at is the dynamics of RPA binding single-stranded DNA, which I know other groups that work on RPA have looked at, but I've not looked into that so far. So whether that's relevant or not, I'm not so sure. But being interested in sort of ubiquitin signaling, I wanted to look at how RPA binding to ZUP1 might modulate or regulate or modify its double activity. So I mentioned the RPA complex could bind to this helix turn helix, immediately N terminal to the catalytic domain. So I wondered whether if RPA requires this domain to bind to it, does this modulate its double activity in some way? And as you can already guess from the title of this talk, it seemed to activate this double activity. So here with just 3 longer case-3-link polybutyne chains, we see that the wild-type ZUP1 in the presence of RPA seems to increase the ability to remove tetraubiquitin and triubiquitin chains, at least in vitro, and over a sort of concentration-dependent manner. Let's put some numbers on this. I've tried to use gel densitometry with a tetraubiquitin chain, and sort of over time, you see increased removal of the longer chain in the presence of RPA, and this is sort of quantified here on the right over several different assays. So we see this increased activity. This still seems to be specific for case-3-link chains. So I've tried it with other homotypic chain linkages of polybutyne chains, and it's still very specific for lysine 63-link chains. And also it's similar in that case when you incubate it with heterotypic chains. So if you have mixed or branched chains, it still seems to have preference for the case-3 linkage to cleave. It seems to be specific to RPA, so I've tried things like prokaryotic single-stranded DNA binding proteins, other mammalian repair replication factors, and it still seems to be just RPA that has this sort of effect in a concentration-dependent manner to activate ZUP1's double activity. And I've tried different orthologs of ZUP1 from mouse and xenopus and plus a few other more exotic members. And when you have this alpha 2,3 present, you still see this enhanced double activity of ZUP1 in the presence of sort of stoichiometric amounts of RPA. And the additional sort of thing as well, that RPA seems to be able to stimulate or activate ZUP1 double activity in the absence of this ubiquitin binding zinc finger, that while it's required for its basal activity, you still see an increase in double activity against polybutyne chains in the presence of RPA, implying that perhaps the mechanisms by which these zinc fingers work and how RPA might activate ZUP1's double activity are sort of slightly separate. And I've tried this with, or tried to get more kinetics or KM information using fluorescence-based activity probes. And here we're not going to be able to get Michaelis-Menten sort of kinetics out, but we can sort of see this increased specific rate of around 10-fold in the presence of RPA for both the full length and the version without the ubiquitin-binding zinc fingers. And using these assays to be able to get apparent or approximate KDs that have matched some of the preliminary SPR and binding data that I've got from early on in the project. So just to finish off, as a working model for ZUP1 RPA interaction, so we see that ZUP1 seems to bind directly to RPA, and this seems to be independent of whether an RPA is bound to single-stranded DNA or not. It seems to require this helix-turn helix domain immediately N-terminal to the catalytic domain. And ZUP1 seems to contact multiple regions within the RPA complex. There was a recent paper from Edwin Anthony looking at RTC105 that used similar sort of techniques. And this seems to be similar to that in that there's quite complex binding of ZUP1 to this sort of quite important replication repair factor. And RPA binding seems to stimulate ZUP1 double activity against very specifically KCD3 linked ubiquitin chains. And our working hypothesis now going into the cell biology aspects is that this interaction is sort of required to recruit a very, very specific dub or chain linkage specific dub to sites of DNA replication stress or DNA damage, either to shut off the signaling or modulate the signaling in some way. And that sort of opens up what the function of it is in DNA repair, because we're looking at ZUP1 knockout cells now, or have been, and we have particular DNA damage types that it seems to be sensitive to, and also sort of leads on to what the substrates of ZUP1. Is it a generic dub for this specific chain type, or are there particular proteins where they are polyubiquitinated that it might act on? And sort of in the next 6 months to a year, hopefully I'll be in a position to present more data on that sort of work. So I'd just like to thank the people involved. So the Gibbs Seymour Lab, so currently it's just me and Ian, but previously we've had Martin as a postdoc, Matt and Kay as DPhil students, and we've had a range of current and former master's students coming through the lab. As far as other people who have been involved in this, so Marjorie has been involved in running them cross-linking mass spec samples, and various other people within the department who provided reagents or access to facilities to get some more information. I'd also like to thank the funders. We're funded by Cancer Research UK, and we've had some internal funds as well. And thank you all for listening. I'm happy to take questions.
Brilliant. Thank you so very much, Ben. That was really interesting. And we will have questions for you later. But right now, let's move to our last speaker today, and that's Eszter Csibra from Imperial College London. Now, Eszter is a synthetic biologist with a background in fundamental molecular and cell biology. And her particular interest is in protein translation and engineering. Now, Eszter completed her bachelor's degree in natural sciences at Cambridge, and then gained her PhD in molecular virology and also in Cambridge, and then moved to UCL to structural and molecular biology department to develop methods for direct evolution of proteins. Now, Eszter is currently a research associate in synthetic biology and is working at Imperial College London. And she's going to talk to us today about the work that she's doing about the absolute protein quantification using fluorescent measurements. And it is called, I think, FPCountR. Unless you want to pronounce it in a more funky way, and I don't know. Super. Well, Eszter, it's all yours.
Thank you very much for the introduction. So, okay. Right, so thank you for the introduction, and thank you for the opportunity to be able to present my work today. As you've heard, my background is in molecular biology, biochemistry, but currently I work in an engineering department in an engineering laboratory on synthetic biology. So the context of my talk is very different, I think, from the previous two talks, even though my background might be similar. So in this context, of course, in microbial synthetic biology, a lot of what we do is engineer constructs and circuits that will do interesting things in E. coli. And the very first thing we typically do when we engineer a new construct, and this includes parts like plasmids, promoters, switches, biosensors, this sort of thing, is to test the expression of the protein in these new plasmid constructs using a microplate reader assay. And this is a pretty traditional thing. So we will grow E. coli in a shallow 96-well plate in about 200 microliter cultures over a period of several hours and watch the E. coli double. And at various intervals, we will measure the absorbance to enumerate the cell count and the fluorescence measurements to enumerate the amount of GFP, M-cherry, etc. in our circuits. And what's the interesting thing in the field is that there is a known problem in doing these sorts of assays, which is that we don't get good units out of this kind of data. In fact, still today, the vast majority of these sorts of assays that are done in the laboratory are presented in RFU, so relative fluorescence units, which are arbitrary units that have no real world meaning. So what this means is we know that, say, if you have twice as much RFU in one sample as another, you have twice as much GFP, but in terms of what numbers of GFP we're looking at in these E. coli cells, we don't know. And in addition, every single plate reader will give us a different RFU, which are essentially arbitrary units. And so in order to compare between different experiments, we have to not only use the same fluorescent protein, etc., but the same plate reader because they're uncomparable. This provides loads of limitations, and this has been a big thing in the field to try to get real units into what we do. What a number of groups have started doing is calibrating their plate readers with fluorescein, which is a small molecule fluorophore of similar spectral range to GFP. And if you make a standard curve of fluorescein, you can start converting your RFU values to molecules of equivalent fluorescein. And this allows you to do all sorts of interesting things, including compare your results with different laboratories or in different plate readers or in different gain settings, so long as every single one has been calibrated the same way. However, as I said, I work with engineers and mathematicians who want to model how circuits work and how cells behave. And for them, it's very, very important that we need molecules of protein, not molecules of fluorescein. It's astonishingly, like, because we don't have any idea about the number of GFPs per cell we express in our circuits, and yet we discuss quite a lot the impact of circuits on the host cell physiology and slowing down host translation and causing host burden. But for the most part, we have no idea even the order of magnitude of GFP that we're expressing in our cells. And in E. coli, this can be any one of 5 different orders of magnitude from 1 through several hundreds of thousands. And we also can't compare between, if we have a GFP and an M-cherry in our circuit, we don't know which one is more abundant. So this is kind of astonishing, and there's a very simple biochemical solution to this, of course, but most people in my field aren't biochemists. So, of course, the solution is purify some fluorescent protein and build a calibration curve based on that fluorescent protein. So if you do this for all the fluorescent proteins in your circuit, suddenly you can have molecules of protein for every single one. But the problem with, I'm not the first one to suggest this, I'm not the first one to try it, but I am the first one to, we are the first ones to sort of do a thorough piece of work on this. And the reason seemingly is that there has been a lot of skepticism in the field as to whether this works, because there are questions of, well, fluorescent protein stability and fluorescent protein purification as a method that a lot of people in the field won't be familiar with and will be too complicated for people who have never purified a protein before. So there are questions of accessibility of the protocol and other challenges to do with accuracy. So when you're thinking about quantitative biology and your need for quantitation is very high, your need for accuracy is also very high. So there's these almost there's 2 challenges of accuracy and accessibility, which are often trade-offs of each other for method development, which are possibly the biggest challenge in trying to get something like this to work and get other people doing this work to adopt it. So what we've done is we've built on existing methods in the synthetic biology literature, which are fluorescein-based methods. So the B. et al. Paper is an iGEM interlaboratory paper that developed a protocol for fluorescein calibration and for using the fluorescein, the conversion factors that you get to interpret your experimental data in units of fluorescein. And on top of this, a paper from Chris Barnes's group developed an R package that sits on top of it and automates all of the analytical processes, both for gaining the conversion factors and for interpreting experimental data. So what we've done is to develop this for proteins. So The major contributions are illustrated here. So first of all, in this case, we're not buying in fluorescein from Sigma and resuspending it. We are saying that everyone can produce their own fluorescent protein calibrants. So we developed a protocol for that. And then, of course, because you're producing your own calibrants, you need to first measure their concentration to relate the concentration to fluorescence to get your conversion factor. So we've been developing methods for accurate protein concentration in high throughput. And then we've made a few other changes that I'll come to at the end on the analytical side. So protein quantification was actually a big part of the challenges that we had to overcome to make this method. So in this case, we're working with purified fluorescent protein. And there are obviously many different ways of quantifying proteins. The most widespread one and the most attractive one initially for us being the A280 assay, which provides a way of essentially turning light absorbance measurements straight directly into absolute concentrations of protein. However, the vast majority of A280 assays in a synthetic biology lab would probably be conducted in single throughput methods, so like in a nanodrop or in a single cuvette. And we wanted something that would work in high throughput in a microwell plate. So we spent quite a lot of the initial study adapting these single throughput methods into a microwell plate. And most of that it's in our paper, it's tucked away in the supplementary, but it was fundamental for everything else. But pretty quickly we realized the A280 assay wasn't good enough for what we needed because it wasn't actually particularly sensitive. We were working with quite low concentrations of proteins. So we developed a new assay that we've called the EC max assay, because of course the A280 is also dependent on an EC, so an extinction coefficient. So this is just a separate extinction coefficient. So unlike most proteins, fluorescent proteins absorb light very well, and they will have a second extinction coefficient at a much higher wavelength than 280, depending on the protein. This sort of varies, of course. And the important thing here is, of course, this extinction coefficient is recorded on FP base. So you can write a script that directly grabs it using the FP base API, which is very useful for our purposes. And it's also important to note that these extinction coefficients are typically 2.5-fold higher than you would have for the A280 assay, which has three major implications. So the first is it's an extremely accurate way of measuring the fluorescent protein concentration. It turns out, because when we compare between the A280 and the EC max, we get very, very close agreement between the two assays. Secondly, it's extremely sensitive because of the fact that the absorbance is typically quite high. So it's not very buffer sensitive, and the linear ranges go down to well below sometimes 1 nanogram per microliter, which is a good 1 to 2 orders of magnitude better sensitivity than a lot of the kits you can buy from Thermo. But the third thing, and probably the most important thing for our purposes, is that because these fluorescent proteins, because these new wavelengths that we're using are quite high, so they're far away from where all the other proteins in bacteria are absorbing, we hypothesized, and it turned out to be true, that we could use crude lysates to actually measure. We could measure the concentration of fluorescent protein even in crude lysate. So previously we were doing expression lysis clarification steps and a purification step to get the pure protein, but it turns out even if we don't do the purification and we look at an absorbance trace of the crude lysates, this is the one in green, you can still see the peak from the overexpressed fluorescent protein, and you can use this peak to work out pretty accurately the concentration of the protein and get very good agreement between the conversion factors from each one, which is very important for us because this allows us to sell the method as something anyone can do. You don't need a protein biochemistry background to do this. So as to everything else, so obviously once you have your conversion factor, you can use it to convert the raw values of the fluorescence that you see into GFP molecules. But there's a few other things that we need to think about because we're working with GFP per cell, so we need to think about cell quantification as well. So the raw RFU data can be normalized and calibrated using this calibration I just described. And the OD600 can also be normalized and calibrated. And this calibration uses a microsphere-based method I don't have time to talk about, but it's previously published in the literature. And we've added a couple of corrections to this because it wasn't working very well when we were comparing between different wavelengths at which you can quantify cells. The other major thing that we added was to add a correction for fluorescence quenching by cells. So we found and quantified that in high concentrations of, when you have high concentrations of bacteria, the bacteria, because they scatter light, they actually reduce the apparent fluorescence of the GFP that's actually inside them. So we added a correction for this. And the third thing that we added that wasn't present in previous analytical protocols is the ability to convert OD600 data not just to cells, but also to cell volume. And what this lets us do is provide a method that can calculate GFP per cell for you, or if you're more interested in it, cellular GFP concentration in nanomolar, micromolar type units. I don't have time to talk about all the applications, but I think they're broad and everyone can imagine what they might be. But here is just one example of a characterization of, so this is an M-cherry plasmid based on 2 SIVA vectors, one of which is medium copy and one of which is high copy. And as you can see, the proteins per cell that you can get from these 2 vectors range from in the range of hundreds of proteins per cell through to hundreds of thousands of proteins per cell. So as I said, there's a huge order of magnitude that it can occupy. And for reference, the average protein in E. coli is expressed in the order of roughly hundreds per cell. Once you get to hundreds of thousands, you're well into the overexpression range in the most abundant proteins per cell. So in this range, if you're expressing your GFP from your plasmid, a vast majority of the resources in the cell will be going towards producing this protein. But we can start looking at quite interesting things when we do this, which is to see that, so for example, depending on which fluorescent protein you use in your vector, you actually end up with different numbers of proteins, which is slightly counterintuitive to some people, because we tend to think that protein expression is dependent on promoters, ribosome binding sites, and these sorts of things. But actually, of course, quite a lot of factors in the translational elongation and post-translational factors can affect how abundant your protein is. And we see this when we swap out M-cherry for MTAG BFP, we see much higher values for the number of proteins per cell. And you can do the same thing, but obviously measure in nanomolar if that's more useful to what you want to do. So in summary, the method is as open as possible because we want to enable as many people as possible to use it if this is useful. So the paper is open access, the protocols are public, and as I say, no longer require protein purification. And the R packages that we built for the analysis is also open source and shouldn't require too much programming experience to get going, because in addition, we've provided some example data and a whole worked example to show you how you might get from RFU to real protein units per cell. So yeah, that's me. I wanted to leave the links up and to put my contact details up. I work in a great lab for a great PI. So I want to thank Guibert Stan and the whole group for being great colleagues, and at Imperial College Centre for Synthetic Biology, which is just an excellent place to do synthetic biology. And yes, so we're funded by the Royal Academy of Engineering. And thank you again for your attention and for listening.
Brilliant. Thank you very much, Eszter, and for a very interesting talk, slightly different than the other one too, but brilliant and very interesting. So now we welcome questions for any of the speakers. If you have a question, please type it in the question box as shown in the image on the screen right now. And whenever you type the question, then I will see and I can read the question to the presenters. So I'll start because we have one question from Ala Osman for Ivan. And Ala is asking whether there are any computational validation methods that can be as accurate as the in vitro or experimental methods used in your project.
Oh, thanks. Yeah, very good question. I mean, spontaneously I could think of. I mean, I don't know if this refers mainly to the structure or so, that if one could uphold the complex structure of the complex, that I've not tried. Maybe that's worth a try. And otherwise, the other experiments, no, I don't. I would not think so. No.
Okay, brilliant. Thank you. Well, because as I'm waiting for other questions to appear, now I'm going to ask things that I'm interested in. So Ben, can I ask you, do you know whether, you hinted at it at the end, but do you know whether RPA interaction help localize ZUP1 to chromatin and to these potentially troubled places during replication?
So yes, I think we've done some preliminary stuff using sort of replication blocks and seeing co-localization in response to those. I think Ian's in the process of doing more cell biology to see making sort of mutants to see if that's dependent on the alpha 2-3 region. But yeah, I think that's the current hypothesis, and we have preliminary work that shows that, but we need to do a bit more cell biology to see if that is the case, particularly at DNA damage. Because from the in vitro work, it seems to bind sort of directly without much regulation. But yeah, how it's regulated in DNA damage, we're not sure.
Okay, and Eszter, can I ask you? So, can you use any type of fluorescent protein, do you think, in your assays, or does it matter whether they monomeric, dimeric, or kind of the mode of action, or how they work?
I mean, the claim is that it should work for any of them. I don't see why it shouldn't. So we tested the three of the most well-used ones. So GFP Mut3, mCherry, and mTag BFP2 deliberately, so they're across the spectrum, and it worked really well for all three. I don't see why it wouldn't work for any of them. If you have one that's particularly unstable for some reason, then that might be challenging. But because we deliberately use E. coli expression strains that are knocked out for certain proteases that would typically make unstable proteins difficult to express, I don't think that's a big limitation. So in theory, yes, any of them.
And Ivan, can I ask, my question, so can I ask, do you, in your research, in your models, it suggests that the APLF is going to bind first and then DNA will come and dislodge it. So can you see that addition of DNA is kind of destabilizing the complex between the octamer and APLF?
Oh yes, yeah. So basically APLF covers only so in principle it covers roughly three quarters of the DNA binding surface on the histones, and then one DNA binding site is free. That is close to the nucleosomal diet on H4, and that's basically what we showed in this one essay, that a small piece of DNA can bind, most likely there, but then as soon as you make the DNA long enough that it can form a nucleosome, then APLF is excluded.
Okay, cool. Thank you. And we have a question from Manish Kushawa for Eszter. And Manish is asking, is the relationship between OD600 and cell density linear, particularly for growing cells?
That's a good question. So yes, it's linear, but only within a certain range. So once it saturates pretty quickly, but for the most part, that doesn't matter too much, in the sense that in a plate reader, because we're using quite low cell densities, or quite low, volumes. Once it saturates, the cells are typically in stationary phase anyway. But this is a well-known limitation of the OD600, but it's currently still the best way of counting cells in a plate reader form that we have. For people who are very interested in getting over this limitation, we typically move to flow cytometry, at which point we don't have this problem, because obviously flow cytometry will count your cells for you, and it's cell by cell data.
Super. Okay, so because as we are getting to 3 p.m., probably the last question is for Ben. Ben, do you think that RPA is the major substrate for ZUP1, or whether do you think that the other replication damage proteins are potential substrates for ZUP1?
So from stuff that's published, there was one figure that showed the knockdown of ZUP1 seemed to increase the level of polybutene chains, possibly on RPA 32 and RPA 70, in response to, I think it was hydroxyurea or DNA damage. But so we're trying to do this in a quite unbiased way now, using sort of ubiquitin binding entities and mass spec and things like that. We have a couple of ideas of what they might be. So PCNA is one that is thought to be polybutylated to lead down to a particular DNA repair pathway. So that's one hypothetical sort of substrate that we think. But we think possibly that this interaction with RPA is to recruit it, and then it's sort of a very specific chain linkage type rather than being substrate specific, given its ability as an endo-dub on longer chains. So I think there might be several different substrates where it's more specific to the chain type.
Okay, brilliant, thank you. Well, thank you very much. Thank you to all of our presenters, and thank you everybody for attending today's webinar. And if you would like to continue this conversation online and ask them more questions, please follow Biochemical Society and Publishing on Twitter. You can see the Twitter handles at the bottom there, so use them and ask them questions. They should follow it too. Okay. Now. Just few organizational things before we close this webinar. So we are really, we would really like to welcome suggestions for future topics and speakers that will be in this series. And you would like more information, please look at the Biochemical Society website when you can submit a very short promise form, with suggestion of the seminar speaker and what subject that you would like to hear about. And please also have a look at the upcoming future webinars. And there is lots of different webinars that are being planned, and there are also the previous webinars which are being recorded, and they are on the Biochemical Society YouTube channel. So please have a look if you would like to watch some of them. Now the next webinar for Biochemical Society is on 23rd of November at 3 p.m. and it is another of the Early Career Researchers webinar series and this one is going to be chaired by Krzysztof Wicher from Macomics, and the seminar is entitled Cellular Behavior in Disease Models. So please have a look if you have time and interest. As you can see from today, they're really interesting. Finally, the last thing is I just wanted to highlight that it's a really good idea to become a member of Biochemical Society, to join the community of people at Biochemical Society, and there's lots of benefits that you can gain from it. From personal point of view, I can tell you that in my career I got 3 different travel fellowships from Biochemical Society. They're brilliant. You can get money and you can put it on your CV later on that you got it. So it actually can get so much more money than you have to pay Biochemical Society to be a member. So it's really good idea to join. So with all of that, thank you everybody. It was a really exciting, really cool subject, really cool talk. So thank you to all of the speakers, and thank you to all of the people that watched us today and asked questions. Thank you very much. Bye.